Halo, cek, cek. Cek. Halo, halo, cek.
Right, test. Good morning, everyone. Okay, so we are really sorry for the delay from the scheduled time. So it was scheduled for 9 a.m., but then it's roughly 9:35 now. So I hope everyone is still excited a little bit about this topic here, about protein crystallography. Okay, so most of the attendance come from the online session, which is on Zoom, basically. That's a little bit like, uh, you're lost on your side there because today's session is mainly like, the Formulatrix 3 team. They've already come over to I3L to really introduce some fancy stuff related to protein crystallography or related to crystallization of the protein molecules. So if you're not here in I3L, then it means that you're not able to come and take a look or to really get some sort of like experience of the hands-on machine that will be shown by the Formulatic team, basically. Okay, so just a little bit of another delay, probably an online delay. First of all, the formulatics team now is still trying to connect to the Zoom meeting and then set up everything. So can you guys see things from the Zoom now? Like the screen has already been shared? Yes. Okay, and I'm audible enough, yeah, for everyone. Okay. Okay, so good morning, everyone. Basically, welcome to the Power Talk today, when we are going to have a discussion about protein crystallization and how crystallography has already shaped, especially protein crystallography has already shaped the development of science, basically. So we are honored today to have a formulatics team coming all the way from Salatiga to Jakarta so that they can start getting to know us in I3L as well, and then trying to introduce about the company profile in formulatics, as well as introducing to us about all these fancy machines that they have already come along, they have already brought along with them from Salatiga. Okay, so for those who are attending here in I3L, you guys probably have already got the hands-on taking a look, but then later on, those who are joining online and then you are still on campus, basically you can just simply come to 208, and then in 208 we will be able to you will be able to get the hands-on experience on the machines that we are that the team has already brought from Salatiga. Anyway, without any further ado, I would like to introduce the team that we have over here. So first of all, it's Pak Taufan.
Yes.
Okay, so Pak Taufan, probably you can start by a little bit of introduction from yourself.
Okay, thank you very much, Mr. Mario. So, hello everyone. Yeah, I'm Taufan. I am informatics working staff from the 2006, and yeah, I'm joining in this company, start coming from the production employee, and then we are start to the doing something the engineering team, and also right now I'm working as the new production manager in informatics. So I will our motivation here, we want to share everything about the technology that we have. We want to share about the what kind of life science information, life science technology that we have, especially for the in the formulatics. And then we want to all of you know about the that technology that can help pull in the scientist research, in the research and development of the scientists. So we are really appreciate and so thankful and working with you guys. And then hopefully the session will be sharing about the information that we can inform to all of you.
All right. Do you want to start first,
Yeah, okay.
Taufan? Okay.
Sure.
All right. So the first session, the first part of the talk today, it's basically about the introduction, a little bit of introduction about formulatrix.
Yes, correct, Mr. Mario.
Okay. And
Correct.
then from there, we're going to continue right away with the second topic of today about protein crystallization. And then I will introduce the talk, the speaker for the next session later on. So
Yeah.
the time is yours now, Taufan.
Thank you, Mr. Mario. So everyone, so I want to sharing about the this is the Formotex building, and Pak Mario already said that we are in Salatiga in Central Java, especially, and I want to share about the Formotex itself is we are some company that focusing into the laboratory automation. We want to make it the life of the scientists easier, and we want to support every experiment that scientists do, especially for the automation life. So why we are doing some automation? Of course, we want to make it work easier, right? From the manually to the automation, it's make us easier. And of course, it's not only about the easier things, but I will to sharing to you about the another thing. And this is just vision and our mission. So our mission is to enable the scientific discoveries in proteomics and genomics for the advancement of the disease-free and hunger-free world. And then our mission is like what happened right now. So we want to collaborate with the research to simplify the preparation and analysis protein nucleic acid by design solution boundaries. And so we want to make it improve the every job of the scientists going better. And we want to make it more doing some automation things that make it life of the scientists getting easier, especially in during the research. So just want to share about the core value why we are grew up until today. It's because we are, of course, this is should be every scientist should has these things. This is the hard work and also the intelligence. And also we have a divergent thinking, sustainability, and as a scientist, as a research and development, we must harness and trust each other. That is the important thing that we need to have to grow up with the formatics. And I believe everyone who want to be the researcher or scientist should have this point. And the next one is I just want to share about the how we are grow in formatics. So we are basically in Indonesia right now. And if you take a look on there, we are in Indonesia. We have our R&D and production. We have a prototyping. We have a testing, and also we have a manufacturing on there. But right now, basically the formatics itself is based on the United States, and our office right now is official office in Dubai, and the base on the United States is more about the proof of concept right now. And then we are doing some research and development in Indonesia. We are doing some improvement of the design in Indonesia. So that's why we want to share everything about this technology that we have right now. So everyone from the research, everyone from here who has some knowledge about the lab science thing can sharing to us. And yeah, this is our location in Salatiga. We have in Semarang in Central Java, and also in Bandung. This is we around maybe 2 until 3 years. We start in Bandung. We call it Formatics Bandung. And then yeah, this is more like our story. So we are founded from 2002. At the moment, we just grew up with the software of the imaging system. And also we making the rock maker and 2005, and then 2006 we start to develop in Indonesia. We start to make manufacturing in Indonesia. And the next information is about the I want to sharing on 2019 in here. So we have a working development in collaboration with QIAGEN at that moment, and then QIAGEN is the big company who want to collaborate with us as a PCR instrument. So after that, QIAGEN acquiring us, so they are like buying our technology for digital PCR. Because our technology in digital PCR is the good things for the QIAGEN itself. That's why they are acquiring on the 2019. So this is the information about our worldwide client, and mostly is we have a pharmaceutical company and some is in university. Yeah, and of course, I hope someday I3L as our client also. Okay, I want to share about this is some like information about our product for the formatics. So we have several categories in here. So protein crystallization, if you can see in front of you right now, we have a rock imager. This is like a more imaging system, automation of the imaging system. And then we have a liquid handling. It's more like how we maintain automation of the dispensing and also aspirating. So we have also working as cell culture. We have a filtration here and laboratory automation. So we call proper. It's more like a automation of the plate transfer during one building into the another building, something like that. And the next I want to share about the because we have two sessions here, and the second session will be talking about the protein crystallography. So protein crystallography will be shared with my colleague, and then I want to share about our technology on the liquid handling. So this is some information that maybe new things to all of you. So why we are working in the liquid handling, because we have several challenging for doing some automation in liquid handling. The first one is about the accuracy and precision, aspirate and dispensing. So usually all of us already know that doing some dispensing using the micro pipette tip, it takes time, right? And then it's what is that? Need several standard operational procedure. Your hand should not touch with the plate, and then you need to change the tip of the pipette every single dispensing. So we are working on that to minimize of that. And then sometimes everyone finds some different viscous liquid that sometimes it's difficult to dispense into the plate. Yeah, of course, will be a problem. And yeah, maybe the last of them on that information is not only the cost, but it's also go green. Go green, something like if you are changing your pipette every time, so how many plastic that must be thrown in this world. Okay. Next, the last one is talking about the cost. Of course, it more if we are talking about the cost, is talking about the efficiency. Okay, the next information is after we are talking with several research in the world, so and hopefully we got this information also from research in Indonesia. So we get that point. So we get that point is the instrument must be reproducible, precise, fast, flexible, simple, and of course automated. And just want to sharing about the what happened on the COVID. As we know that on the COVID-19, everyone need to be going faster to doing something. So that's why some of our product in liquid dispensing, some of technology in liquid dispensing is really helpful for the scientists, make it some sample, or maybe the researcher doing some testing of the PCR test. Because usually from manual hand going to the automation. Okay, so the next one I want to share about our technology that we have. So in the liquid handling technology in formulatrix, so I want to share to all of you about the microfluidic pump technology. So the microfluidic pump is, if you take a look on that picture, so it we call in formulatrix, usually we call chip on there, and inside of it it has some silicon membrane on there. So the silicon membrane or perfluous membrane that can be used as a diaphragm for pumping the air. Maybe it's too hard for imaging the what I said about the microfluidic pumping. I want to share about the video to all of you. Yeah. This one is the video of the microfluidic pump, how we working with microfluidic. If you take a look on inside of it, inside of the chip, the tools for dispensing, so it has some like yeah, we call it a microfluidic valve. It created from silicon piece. So how it works, it's more like this. So yeah, every single part of the silicon, it has some function. So the first function is taking the liquid from the original sample. It can be from the pipette tip, it can be from the bottle, and etc. And then the large or small diaphragm will be pumping into the some plate. So it working as a like a valve, or what is that? We call a valve cluster or valve output in input. And then after the process, so if you take a look on the picture right now, on the video right now, so the large diaphragm getting what that working by sucking the liquid, and then the output valve start to be open, and then the next one is the diaphragm are, because the diaphragm are closed, it has some pressure on there, so that's why it can dispense the liquid into the plate, or in some sample into the plate. So there is the how we working with the plastic. Sorry, I mean silicon piece. So because the elasticity of the silicon, we can make some membrane like that for dispensing some liquid. If you take a look on there, it can be adjustable by what is that? The volume that we want to dispense. And yeah, it can be, you can imagine if there is the pumping going faster with the time, with the faster time, so the dispense will be faster. Okay. Yeah, the next information is about this is the another technology that we have on in life science automation. There is if you take a look on the which one? My left, my right side. Yeah, my right side. It has some conventional tips. Yeah, all of you, I believe you know about the conventional tip. If you take a look on the conventional tip, people always talking about the how much precise or how much accurate for the pipetting on there. So that's why because there is a have some like air cushion on there. We call air cushion on there. You see the gap of the between the liquid and then the end of the micropipette tip. But if you take a look in the left side, this is some another technology that we have in the automation. We call a positive displacement pipetting. So in the positive displacement pipetting, if you imagine, there is like more like a shearing. So it has some what is that tip? There is have some axial seal on there. So we are really minimize any air cushion. So there is no air gap on there. So that's why it's more precise. Because of that, we can dispense with the volume range around 100 nanoliters. So how it the next technology is about the conductive tips. So maybe this is the new things on the technology about the conductive tips, because the conductive tip it has the differences between the conventional tips is the conductive tip has something like we call a conductive strip on there. So if you see the black line on there, so there is the conductive strip. It can what's that? Detect the height of the liquid. So if the liquid are low, so the tip will be going automatically following the size of the liquid. So the video will be like this. So if you take a look on the positive displacement, so we call mini shearing on there. So it's on the left, on the right side, and then the left one is the conductive tips. So in the positive displacement, you can see there is some like a small shearing on there, and then just doing some aspirate, and then doing some dispensing again. And if you take a look on the conductive tip, so that's why it has some conduction on their conductor on the tips itself. So the tip can follow with the size of the liquid on the plate. So there is the conductive tips. So yeah, it's more like following how the height of the tip. Of the liquid. Yeah, the next one is I want to share about this is the next generation we call a flexible finger. If you take a look on this one, so this is the surface of the plate is not flat enough. So that's why sometimes the research or scientists find some difficulties when they want to dispense by using automation. But this is the another technology that we have for making the dispensing more flexible to get some liquid drops like this. And this is some we call drop dispensing because this can be used for the protein crystallography research. Yeah, by using the flexible finger, of course the liquid that dispense it's more precise on there. And the next one, we want to sharing about the I just share about the go green before. So this is some the example that we have in our technology. So mostly everyone just throw the plastic pipette after they are used. But this generation, this technology, we are making some some tips that still we can still using that tip by washing it. So it's more make it of course it more make it like we don't throw it the plastic for every research. Yeah, this is the example that I want to share about the how we maintain the cost by using some automation instrument, by using some automation technology. So we can make some like miniaturization reaction. What is the miniaturization reaction? So there is we more like usually for the standard of the company, they have some standard for doing some pipetting into the some sample. And of course, if we are talking about the cost, just imagine if the reaction it so high with the price is so high, and we just following the standard operational procedure from the vendors, from the someone who produced that, so it will be create the highest cost of there. So by using the reaction miniaturization, we can like divide the standard operational procedure from the vendors. And because in the automation technology, some automation can dispense with the precise accuracy with a good accuracy for dispensing. So that's why we can divide it the standard value. So it's more like make it more less dispense or less drop on there. That's why just imagine if in some standard operational procedure we can dispense with more volume. So by using automation, so it can divide it, and of course it can be reduce our cost. So this is some example that we are talking about the our technology for the pipette conductive, and also the positive displacement in our formulatrix technology. And the next one, if you take a look in front of you, it has some we call mantis on there, because this is the single dispense to doing some auto dispensing, and it can be dispensed until 100 nanoliter. Okay, so when we are when COVID-19 happened, so formulatrix helping several scientists to do some research doing when COVID-19. This is the information that I want to share to all of you, because in automation dispensing can reduce the reagent consumption and can make it faster as a dispensing solution. So that's why every scientist, when they are doing some research related with the COVID, they got some interested with our device or this technology. So this is our activity in Formulatrix Indonesia. So we are doing some like PCR test by ourselves with using our instrument that we have. So we are using Mantis on there. We are using some several instrument of liquid handling. Just want to learning about how this technology can be released into the world, into the life science equipment. And not only that, we are during the COVID-19 in Salatiga, we are working with the we call a balai vector on there. So it's more like some institution that working with government for COVID activity, for checking the PCR test. And yeah, if you take a look on there, we have they has some 2% as a scientist on there, and they will suffer for doing some manual dispense, and it takes 13 minutes to make it some sample, and it will be take it more so stressful on there, because as you know, during the COVID, that's why the PCR test cannot release in quickly, because there is so manual on there. So we are working with the this institution for our Mantis, for our automation dispensing on there, and they can dispense. They can using this automation device for their lab, and they feel so helpful with this automation, because before they are using the manual, and right now they are using automation device, and it only takes 1 until 2 minutes for dispensing for each sample, for one plate. Sorry, not each sample, but 96 samples. Okay, the next one is I want to share about the how we live with Formulatrix. So this is our life in Formulatrix. So in Formulatrix we have a R&D team. So we have a mechanical, we have a process engineer, we have a software, and we also have some scientists on there. So yeah, we are trying to how we improve the technology for everyone. We want to improve this technology to helping scientists and research team doing some how it can make their life easy. And yeah, we also have a production team in Formulatrix who working with the several department on there. And yeah, this is some information to all of you that Formulatrix Indonesia is really open with the educational institution to having some collaboration. So usually we have some collaboration in here related with the research, thesis, internship program, and also recruitment. And also yeah, this is some example that we are doing right now. So we can be a speaker, of course, to the some educational institution. And also yeah, we really hope that I3L will be part of us for this collaboration, doing for some, yeah, anything of course that related with education. Yeah, and the last one is I just want to share to all of you that this is some educational institution that already working with us. We have Satya Wacana University, we have Olban, we have some several institution on there, and it's working start from the mechanical until the science. Yeah, this is I think hopefully can be start for us that we want to collaborate with all of you in here. So hopefully I3L and Formatrix can collaborate to make it improvement for everything about the knowledge of the automation. Of course, it will be our improvement how we can improve the and sharing our technology to all of you, and hopefully the member of the I3L can get some what is that like can get some information or sharing some information about the what should be Formatrix do for life science industry, something like that. Okay, I think that's all from us. Thank you for everyone. This is the introduction for you, and I just want to share about the technology that we have in Formatrix, and hopefully this is can be a start for all of you to know about the what kind of the technology on the automation on the life science things. So there is the several technology that we have in the world. So thank you very much and appreciate to all of you. Mr. Mario.
Okay, thank you, Pak Taufan. Okay, so everyone, there was just like a little bit of introduction about Formulatrix, and then now, like we've got a person over here. Actually, unfortunately, we don't really have the ones from cohort 2018, 2019, or 2020 or 2021. Probably, if you are interested in like getting to know about Formulatrix even more, say for example, like talking directly to the person in charge from Formulatrix, if you're interested about joining them as an intern, okay, that can also be something that you can do. Unfortunately, you don't come or you're attending it online, so that's your loss anyway. All right. Another thing that Formulatrix team has already brought over here, they've already brought all these machines. The one that Pat Taufan has already mentioned before about automation. If you guys have already worked in the lab with 96 well plate, when you have to start like withdrawing the liquid using the pipette tips, which has like 6 or 8 or even 9 heads, the difficulties when you have to like press the tips and then try to make sure that one of the tip is not going to fall down, and then every now and then you have to like keep repeating the same thing again. Once you withdraw it again, and then the liquid has already been inside, and then the tip is falling down. It's just like so painful when you're doing that kind of work. Okay, and then Formulatrix is bringing this equipment over here where they kind of like make everything in automatic process, right? So they kind of like showed it to us at the very beginning of the session, and then it's just like wow, okay, this is something that probably later in the future you guys will encounter as like as a researcher when you're doing your own research in the future. But then again, that's your loss because you join it online. All right. But then if you are able to come, or like after the class or after the session, like Formulatrix stream will still be here up to around 11 a.m. or something like that. Yeah, Pak?
Yeah.
And then feel free to come to 208, and then you will be able to take a look right away. Especially the main thing for today is about protein crystallography, which is going to like they have already brought this fancy machine as well for visualization, and then the visualization of protein crystals. For someone who has been working with protein crystals in the past, you really feel like it's almost similar when you're trying to take a look on microorganisms under a microscope. When you need to use different kind of like lenses range 40 times, 100 times, etc. Right. So with protein crystals, you have to make sure that you can identify which one is protein crystals and which one is salt crystal, right? And then this machine here is allowing us to not really use that old technique of using microscope anymore, but basically just simply put your 96 well plate containing the protein crystals. Take a look under that. It's been connected to this laptop over here with the visualization software, and then everything is going to, like, according to the number of the plate or of the well that you're looking for, A1, A2, A3, you can just always arrange it, and then everything is going to appear there. And then you can also use the visual light wavelength, or you can also use the UV wavelength. Okay, so that's what Formulatrix team is trying to introduce to us here in I3L and to you guys, the I3L students, because automation in the lab in the future is going to be, like, something that we will probably need. Okay, do we have any questions, by the way, for Pak Taufan? From audience here, Kenneth, Frankie, and Peter. Peter. Okay. So Peter, do you have any questions? No? Or how do you choose your wavelength? In the protein crystallization, Pak Taufan, you want to?
So yeah, related with the specification of the Rocky Major. So it has some wavelength on there. So it controlled by software in here. So we can control by the wavelength. Yeah, it has some filter on there, and then we can just choosing the wavelength inside of the option of the Rocky Major.
So all you need to do is just click on the software. The interface is over there on the laptop. You just simply have to choose visible, click on the visible, click on the button on UV, and then the UV, remember, biochemistry 101. All right, so there are 3 amino acids which are going to be visible under UV light, okay, 280. All right, phenylalanine is one of them. As long as your protein molecule has already crystallized and the protein molecules containing have those 3 amino acids, then it means that under the UV light, it's going to show, it's going to appear something that looks like a little bit whitish on the visualization. So you can be certain that, oh, okay, that looks like crystal. And then that crystal has a strong UV spectrum being shown. Then you can be certain that, okay, that's a protein crystal. Okay, so that's one method that you can actually do on protein crystallization.
Yeah, and of course by the software it can be we can know the signal of the wavelength, so we can control everything.
Okay, go. Any other questions? Maybe from the audience in the Zoom? No? No questions? Okay, cool. Thank you very much, Pak Taufan.
Thank you very much
for the introduction about Formulatrix. Yeah. Okay, the next session, this is going to be the main thing for today. We have our next speaker, Miss Inge. She is actually our own graduate. She graduated from I3L, and then she's now working with the protein crystallization, or basically it's more about, like, trying to introduce the visualization machine, which has already been brought here all the way from Salatiga to I3L. Okay. And today she is going to explain to us a little bit of introduction about protein crystallography or protein crystallization itself. If you are from biomedicine, pharmacy, as well as biotechnology, you will have a little bit of introduction on XRT, X-ray crystallography on one course which we call as analytical instrument and bioassay. Okay, so in that course there will be only, like, one session related to X-ray crystallography. But today, Miss Inge here is going to really explain very thoroughly about protein crystallography in general, and then it's going to be hopefully it's going to, like, enlighten you guys even more and enrich your knowledge about protein crystallography. And then if you have some sort of like, technical questions related to the machines which is being brought here, that you can always like, again, they will be here up to 11 a.m. today or 11:30. You can always come to 208 and then try to talk to the team over here. Okay, without any further ado, Ms. Inge, all is yours.
Okay, so thank you, Mr. Rego. Okay, so to talk like itself, I'm Ina and I'm currently working as a lab scientist in manufacturing Indonesia. And I was actually an idea of Okay, so I'm Inga and I'm currently working as a QC lab scientist at Pro Manufacture Indonesia, or we call it as Formulatics Indonesia. And I'm actually an IPL alumni from PT15. So I think from the one over here is which major are you? PM. So all of four of you are PM. Oh, you have one PT here. Which cohort are you? 23. Wow, I'm that older. I'm really that old. Like I'm 7 years older. Yeah, I know, right? Yeah. But anyway, so thank you so much for the time for today. And before we start, I would like to show you something cool. So I'm gonna run this machine first before we start. Then we will later see the result at the end of the presentations. Okay. So wait. Okay, later we will find out. Okay. Okay. Okay, while waiting for the machines to actually work, let's move on to the presentations itself. So basically today, I'm going to talk more on the protein crystallography itself, starting from the crystallization process. And by the end of the stations, we are hoping for you to actually understand how the sample preparations for the crystallography actually needs to be prepared, how the crystallizations occurs, how you can actually visualize those crystals under this microscope. So first things first, let's talk about the protein itself. So maybe if you are like the first year, like maybe you got like the biochem, maybe like you've learned something about it. So this proteins is actually like the building blocks of our body. So basically like all of your body is comprises of protein. And the smallest unit is actually the amino acids. So if you ever heard like the amino acid, so we actually have like the carbon group here, you have the hydrogen amino group, and then the carboxyl, and you have this R group, which we call as a functional group, or someone also call it as a prosthetic groups. And this group actually give the differences between those existing 20 amino acids. So this is like a chemistry 101. So as you can see here, this is like the structure of those 20 amino acids. So let's see here, we have a serine, you have this basic structure, and you have additional carbon over here and a hydroxyl group. For the aromatic ring, let's say we have like the basic structure here, then you will have like this carbon and another aromatic here. So these 20 different amino acids have different molecular weight. They have like different dissociation constant. They have different isoelectric point, which we'll get into it later. And these are actually very important to the structure determinations and the functions of the protein itself. So there are like very broad functions of the proteins in our body, starting from like the basic one is actually the enzymes, like the trypsins, which actually helps us to digest our protein inside our digestive systems. We have also polymerases, which I believe like all of you know that this enzyme is very critical for our DNA applications. We also have like different like these receptors, and even the muscles that you use to actually move is also comprises of protein. Yeah, let's take a look at the structure of the protein itself, starting from the basic one, which is like the primary structures. So this is actually when the amino acids just binding, like forming a long chain, which we call as polypeptide chain. And then when they interact with each other, like say the A1 interact with A2, they will create these secondary structures, which we call like the alpha helix, or they can also mix like cleavages like here. And when these two different secondary structures, then they will interact again. They will make a bigger structure. They will have like this tertiary. They fold again. We perform like making a complex. Then we will have the quaternary structures. So this is actually the proteins that actually works in the body. So let's say we have like this. This is a hemoglobin. They have like this whole structures with 4 heme group, which later will bind to the oxygens in your body. Now, talking about the structure itself for like the BD students, that this is like one of the most critical things that you need to know, because actually the structures actually determines the functions and the interactions between those molecules. So let's say over here, I have like the blue one are receptors, and then this one on the left we have like 4, let's say 4 drug candidates, which later will bind to different sites of the receptors. So by binding this, let's say we have like this drug A, it will directly bind to this site, and it will produce like a mediocre effect. While the one with the D, which bind to the other side, will only produce like a subtle effect. So by knowing the structures and knowing where to bind, you can actually maximize the functions, let's say like the drugs, and to maximize the interactions between the protein itself, which is why it is very important for you to understand and to determine the structure of the protein itself. So there are different methods to actually determine the protein structures. Like the first one, which actually we are going to talk more about today, is like the X-ray crystallography. So this is like the old method where, if you remember the first one, when the Watson and Crick try to find out the double helix structure, they also actually use this kind of X-ray. And you can also use the NMR. Basically, but this NMR is not only for protein, they also to define different chemical structures, because they basically define like how many carbons you have, like the bonding, is it a double bond, triple bond, or a single bond. And they also have like this cryo-electron microscopy. So this is like a rising method actually, because it's getting like widely used now. So it's basically like an electron microscopy, but with cryo samples, and you will actually be able to observe molecules with like even smaller size. These 3 methods have different advantages and disadvantages. So let's say when the X-ray is actually have like the highest resolutions, but we have a bottleneck where it is very hard to actually find the crystals. To make the crystal itself is very hard. Well, if we take a look at like the NMR, or like the NMR, or like cryo-EM, the sample preparation is much actually easier for you. But in terms of the target molecules, they are very limited. So that's why people are still trying to use this X-ray, because it has like the highest resolutions, and you even can go until like the detailed atomic informations we can go from the X-ray crystallography. Okay, so let's take a look at the statistic from the protein data bank right now. So mostly they are using the X-ray crystallography. Well, as I told you before, for the cryo-EM is still rising. Maybe at some point it's true that it's going to like surpass, but currently the most well-known and like the validated method is mostly like the X-ray crystallography. Okay, so let's go to the basic of the X-ray crystallography itself. So it works by the principles that the crystals will diffract X-ray to a specific directions. So let's say you will have a crystals over here, X-ray coming this way, it will diffract to a certain directions. So these diffractions will create this, what we call as a diffraction map. So when you have a crystals, you will have like an amplified signal. So the crystal itself is actually basically a regular repeating unit of cells. So you can see that this unit cells is repeating each other, and it's a very ordered way, which is why we can actually see the diffraction pattern. So remember, if you only have like a random solid protein, then you will not be able to see those diffraction patterns. Because that's why we need to have like this beautiful crystals to actually see those patterns. But in real life, it's not that easy to actually obtain those crystals. There are like several criteria that you need to obtain to actually have a good crystal. The first one is to actually have a high purity. So basically like a purity of 95% or above is ideal for you to work with. And then the concentrated samples, usually like 5 to 10. Some scientists also use it like up to 25. It really depends on your protein. And we will like to have like a perfect crystals. So it should be like nice, no cracks, no inclusions. Let's say when you have like the crystallizations, then suddenly you have bubbles. That's a big no. Okay, so let's start with the workflow of the protein crystallography itself. So the first one starts with the protein purifications, which we try to actually obtain only the protein of interest. So let's say you have a pool of proteins that we are going to isolate only one of them. And then after you got the purified proteins, you will go with the crystallizations. And after you got the crystals that you really wanted, then you can go with the diffractions and the data analysis. And we will go through this one by one. Okay, so chromatography. So I believe like some of you actually knows this chromatography method. Like this is one of the most employed method to actually purify protein. So basically this chromatography method utilizes the characteristics of the protein itself, how it interacts with the stationary phase of the column chromatography. So in a chromatography, we usually have like this tubes, and then you have the stationary phase, which is like a separator, I would say. Then you will put your sample into it. It gets separated inside the stationary phase, and then it will get eluted one by one. Okay, let's get a little bit into details. These are like the three mostly used method of chromatography. The first one is the gel filtration. Someone also say call this as a size exclusion. So basically you have this like a polymer gel bit, and it has like small pores. So when you put your protein inside this, the big size will go out first, while the small one will get trapped inside the pores of this gel. So it will get diluted at the end. Another example is actually an affinity chromatography. So this is mostly like a specific binding. So usually they use like an antibody to actually specifically bind to your target protein. So they have like this polymers, and you have antibody attached to it. Then you put your samples in. So your protein will get attached to this ligand, and later they will get eluted at the fractions that we wanted. And another one, we have also like this ion exchange chromatography. So as protein have different charges, especially like at different pH, so we are going to try to utilize that to actually obtain the protein of interest. So let's say when you have a positively charged protein, then maybe you will go with a stationary phase, which is actually a negative one. So those positively charged protein will bind to the negatively charged stationary phase. That's how. So this is like the most commonly used method in protein purifications. And actually, these all systems are actually can be automated. And I believe we actually have a Naxta start here, right? Maybe I think it's on the third floor at my time. So we are actually using this. So you can actually use this to actually purify your protein. Another method to actually purify your protein is actually through ultrafiltrations and diafiltrations. So this is particularly important because let's say when we purify our protein through those columns, let's say we dilute it in like a phosphate buffer. So this phosphate buffer is actually not ideal for crystallization. So you need to exchange the buffer. So we can use this ultrafiltrations diafiltration method to actually exchange those buffers. And since we need to obtain a certain concentrations of protein that we want, we need to do a concentration stats. There are different methods to do this. Like one of the two of the most widely used method in the lab is actually a centrifugal filter. Maybe some of you have seen this because if we buy those kits like the DNA extraction kit or RNA extraction kit, you can actually have like a microtubes, and you have an additional tubes with membrane at the bottom of the tubes. And this is just basically you just put in your samples, Then you centrifuge it with the regular centrifuge, and those molecules will get separated based on the membrane size. Or the other method that is widely used not only in the lab, but also at the manufacturing setting, which is also what called as a tangent flow filtrations. So you will have like a filter over here, and then you have a pump, you have like pressure gauge, and so many setup to actually get into this. And this filter is actually quite different from the regular filters that you use to sterilize your, let's say, your media, which has like a pore size of 0.2 microns, because they are like very small. The size is in kilodalton usually. So there are different sizes that is commonly available in the market, like the 5 kDa, 10 kDa, 30 kDa, and etc. So this is like the difference of the centrifugal and the what we call as a tangential flow. So let's say when you have this membrane, the flow of the liquid is actually perpendicular to the membrane itself. So it will go down. So those molecules that has a size smaller than the membrane will directly go down. While the ones that is bigger than your membrane size will remain at the upstream side of the filter. If while you're working with the TFF, the flow of the liquid is actually parallel to the membrane itself. So it will go, instead of going downwards, it will go sideways here. So those have like, say we have like a 10 kDa. So when you have a protein larger than 10 kDa, it will go sideways. Well, like the other molecules will go down. So it's just like a matter of directions of the flow. And of course, it has like its own advantages and disadvantages. So when you're working with like a small volume sample, let's say you have only like 2 mils, then just go for the centrifugal one. This is gonna be much easier. But if you have like a large volume, let's say you have like 50 mil, this is gonna be a problem. Because like all the molecules will be written here, so the flow to go down, it will be much slower, and sometimes it got blocked. So you can actually have no filtration flux to go down like that. And the one that I started before is actually a type of TFF. Wait. Okay. So actually, the liquid that I have on the left side is actually a mixture of milk and food coloring. So this is actually a mixture of milk and food coloring. I have like 50 mils at the beginning. So we have like 50 mils. Then I'm going to concentrate it. So those molecules, oh, by the way, I'm using this 10 kDa filter. So the protein will remain here, while those like the food coloring, the water, will all come out through this side. Like this, because it flows that way. So it will go parallelly through the membranes and it will get separated. So this is what we usually call as a retentate, because we will retain this. This is the one that we want to keep. We call this as a permeate. So this is the one that we are going to throw away. Maybe later, if you want to come closer, we can get it later. Oh, maybe you can really see, right? It's really different because this is still clear. It's only water and food coloring, while all the proteins went on this side. Okay, so let's move on. Oh, I think we can skip this. Okay. So once you actually got the protein concentrated, you might actually want to check first if this protein actually has reached the concentrations that you wanted, or you might actually want to dilute it, or you want to concentrate it more. So there are different methods to actually measure those protein concentrations. The easiest one is actually to have like the with the spectrophotometer at 280. What we call here we have nanodrop. So basically, you just can put like 1 or 2 microliters and you close it, and then you will get the up. It's very easy. It's very straightforward, and it's the principle of this machine is that actually those aromatic ring amino acid they are UV excited at 280. So that's why we can get those readings or the absorbance readings using this nanodrop. The other method of the protein concentrations determination is actually a BCA assay. So this is what we call like a bicarbonic acid assay. So we have different components on the reagent. We have like the main star here is the bicarbonic acid. Then we have a copper sulfate pentahydrate, like we can do any hydrate actually. So basically you need just copper sulfate. So what really happens here is that your protein will then interact with the copper. This copper will be reduced and then it will react with the bicarbonic acid. And this is what actually makes it colors purple. And based on these reactions between the copper ion and the bicarbonic acid, you will get this nice purple color. So like this is our experiment. So we have like different concentrations of proteins. So if you get like a more intense purple, it means that you have a higher protein concentrations. When you have like a very light purple color, it means that you have like a very low protein. And this is actually a spectrophotometric assay as well, because you need to read this absorbance at 562 nanometers. And of course, these two methods has their own pros and cons. So let's say when you're working with the nanodrop, it's very easy. You don't need to do like different kind of reaction. Just take your samples, put 1 or 2 micro in, then that's it. But in terms when you are working with the PCA assay, then you might want to work with like, oh, I need all the reagents, all those stuff. But when you have like let's say when you are having a protein solutions, which can actually have like an emission or absorbance at 280, then it's gonna be a problem. So you might not want to go with the UV method, but you should go with the PCA assay. So there is no right or wrong method in determining your protein concentrations. It really depends on your like sample volume, the solutions of the proteins that you have. Because if you are also working with those like let's say you have a buffer with EDTA or like the magnesium, then you will not be able to do with the PCA assay. Okay, so now let's get deeper. Once you got all the requirements like a purified protein and a concentrated protein, we will go with the crystallization itself. So here we have like a graph, like a protein concentrations against the precipitant. And at the beginning or the solutions that you have is actually at this stage. So it's undersaturated. So it's very stable. It's fully dissolved in your solutions. And over time, as the solutions evaporate, it will go through this solubility line going to that side. So this is how these crystals are formed. So once you pass the solubility line, you will enter what we call as metastable zone. So this is actually like one of the places which actually favorable for your crystal growth. And usually this two metastable and the nucleation zone is actually favorable for you. So during the nucleation zone, you actually have like a microcrystals, but actually the crystals, the growth is not as fast as those in this metastable zone. So usually people are working at these two zones, between these two zones. But if you go over like the solutions, the concentrations maybe is too big, it will go to the precipitation zone. So it's not crystallized, it's just a random solidification of a protein. You will not be able to get the pretty crystals that you really want. So this is why we are maintaining those protein to be in this two phase and not going that side. Later, during the visualizations, we will be able to tell which are the precipitations and which are the crystals. Okay. So now that we are actually in a very modern world, there are actually so many different kinds of protein crystallization kits available, like from the Hampton research, the molecular dimensions. But these kits are actually quite different from the one that we use, like the DNA extractions or RNA extractions, because those are ready to use. You just mix it and you will be like 95%, 100% works. But that's not gonna work with this protein crystallization kit, because you still need to find the suitable conditions for your protein crystal to grow. So it's a kit, it's easy for you, but you still need like several optimizations to find the most favorable conditions for your protein to grow. So this is like an example of a crystallization kit that is available. So you usually have with this buffer and precipitant, then you still need to have like a different conditions to actually define which condition is favorable for your protein crystal to grow. So you will like, let's say we have like a sodium acetate buffer over here, and then we have the precipitant is the sodium chloride. So we're going to make the concentrations and the pH here a bit different, like 4, 4.4, 4.8, 5.2. And you will also be able to like modify the concentrations of the sodium chloride. So this is how you screen what we call as the screening to actually define the optimum conditions of the protein crystal to grow. So it's to the fact that actually protein is very sensitive. So it's in different kind of environment, like in different pH, different concentrations, salt concentrations, it will behave different. That is why you need to do this kind of optimization. And for the method itself, yeah, now you already know that the solutions that you need to have, you need to know the method that you want to go with. The first one is the sitting drop method. So you have a special well. So it's actually the well like the MRC plate. Later you will see it over here. So it has like a sitting, this like a pedestal above the reservoir. So you just put your liquid in and then just fill up. Then you will let it to like evaporate and the crystal will form around here. Or the other, another method that is widely used is what we call as the hanging drop method. So basically you have like a regular plate, but we can use like the VXPM plate or Lindbrook plate, which in terms of size is much bigger. So you have the reservoir at the bottom. You have a cover slide and you put your proteins on the cover slide. Because it's hanging like this, it will be hanging. Or you can also have like this microdialysis method in which you are using a semi-permeable membrane. So you have the protein here, you have the semi-permeable membrane, and you have the reservoir over here. And because like the saturations, they will like transfer molecules between this semi-permeable membrane and the crystal will form. Okay, so to get a clearer, better understanding on this, I have a video of a hanging drop experiment. Okay, so we are using a 24 well VDXM plate, and we are going to have like the hanging drop. So we usually use like the isopropanol. So this is just to let the cover slides attach to the adapters. So we have like the cover slides over there. Okay, so once you have like all your cover slips there, then you can actually pipette your protein to the cover slips like this. So this is using our instrument. So we are like pipetting small volume of the buffer first, and then we're going to put it on top of the cover slips, and we will mix it with the protein of interest. And after that, we can just like attach the cover slips to the plate. So basically, this is how you set up a hanging drop experiment. Like this. So you see like the black dots at the top of the cover slip is the proteins that you want to crystallize. So this is like the type of crystallization plate that you want to have. So this is like the one that I said, like the MRC. So you have like this is the reservoir. And you have two dots over here is actually the one that you're going to put your samples into. And this is just like the hanging drop. So it's more like similar to the 96 well plate, but it's just a bit bigger. Yes. Okay. So now that we have like go with the crystallization process, and now it's actually the hardest, another challenging part, which is also actually like challenging for all the scientists, and it takes very a long time to actually screen your protein of interest. So usually the conventional method is to use like the regular microscope. So basically you can use the regular microscope to actually see which protein is actually which crystals is actually your protein of interest. And there are also like nowadays we have like different automations. Like the one that we have is the rock imager over here. And the difference is that we have like different automations. So you will be able to see directly like this. We have like the screen, so you can just easily see the image of the protein of your interest. And actually the crystal, if it is successful, it looks really nice. It looks very pretty. Let's say you have like a very clear, like this needles, like structure, all this like a cubic face. But in real life, this does not work. This is what you usually find when you do your research. So you will, let's say, when you did like a bed pipetting, then you will have like this, like all the solutions will like get split up, and then you have only like a clear solutions. You basically, this is when you have like when your protein concentration is too low, you might actually like expect something like this. Or this is what we call as a heavy precipitate. So this is like when your protein is like all the lipids is evaporated, you find like a solid structures, but it's actually not a crystals. So basically it's like oversaturated, like very, very saturated. You have like a bright precipitate and phase separations. This is also affected by different protein concentrations and the conditions of the screen cell. And the one that we want to see is the one at the below. And this, like the small crystal, microcrystal, it's even harder for you to actually see and really determine that this is your protein of interest. And that's the challenging part. So this is the continuations of the previous slide. So let's say the first 24 plates that I have, this is like the after 20 hours, we can see like several crystals, but not on all plates, which means that you have like the optimum conditions around this area. So later you will optimize more on this side. And this is actually the manual scoring. Let's say this is, I took this from the Hampton research. So they said like, if you want to score your protein, you can go with these classifications. Like if you already have like, see a clear drops, then it scores 1. If you see phase separation, that means the score is 2. And the higher the score, it means that you are most likely to be successful to get your protein crystal. And again, this challenge is that this limited visibility. So let's say you may not see that. Let's say this tree actually has like a very small crystals. And what if at this tree B actually has a micro crystals? So you will not be able to see it with like the regular micro. And it's even harder when you have like this salt, like the sodium acetate, and then you have the sodium chloride. They can also perform crystals. So that's why you may like meet like a fake protein crystals. So maybe you will work with the salt crystals instead of the protein crystals that you want. And that's what Formatics trying to help. So we come up with this technology what we call as a rock imager. So for the one that actually comes here can see it clearly, we have like this imaging like the microscope, and this is actually our bigger version of our rock imager. It has like the storage itself, and they will take it from the storage area, and then they will image it directly under the microscope that is attached to the machine. And not only this, we can actually have like an automatic scoring of the crystals. So this will help a lot to define like which one are the most optimum conditions that you want to have. So let's say those red, you can just neglect it, and maybe you can focus more on those green scoring. And it will be much easier for you to find the optimum conditions of the protein crystals. Okay, so now let's get deeper into the visualization process. So let's say, I think before Mr. Mario already mentions that we can have like different visible lights, UV lights, and those lights actually help us to differentiate whether it is true protein or it just like the fake one. So we have like different light configurations which can help us. Like the first one is actually the visible light. This is the regular light. So you can actually see these pretty crystals under microscopes. And this is but this method has limitations which you cannot actually differentiate between the protein crystals and the fake crystals of the salts that you can find in plastic. Well, if you have a UV light, your life will be much easier because those aromatic groups can be excited by UV. So you can see clearly that this round shape is not your protein. It's only like from salt. So you will only have this, that this is your truly protein crystal. But the problem is, what if your protein has a very low dose aromatic group? So maybe you may not be able to see this nice picture. Then we can have another options. So you can have actually your protein with a fluorescence. You can have a protein type fluorescence. So we have like a different wavelength of light. We have the UV, and then we have those like the yellow and the red one. So you can have like a different fluorescence tag to your protein. So let's say on this side, when you see under the visible light, you will only see like a precipitate. It's not nice, but when you see it under the multi-fluorescence imaging, you might actually see that there is a hidden crystals of it. And this is what usually missed by the scientists when they are using the conventional method. Or you can also see that there are different types of protein, or if it is like a protein complex. So we have like this, the bovine pancreatic trypsin inhibitor, which actually one of them binds to the fluorescence and the other binds to the Texas red. So if you see that those crystals lights on both imaging wavelength, it means that it's actually a protein complex. It's not only just a protein, but you can also have a protein complex of it. Another one, this is like a more sophisticated method, which actually have like a bigger or higher accuracy. This is what we call as the second order nonlinear imaging of chiral crystals or sonic. This is actually a combination between the second harmonic generations and the ultraviolet to photon excited fluorescence. So what it works is that this second harmonic generations is able to detect a chiral crystals. So let's say when it's protein, but it's not crystals, then it will not be excited during this visualization. So it has this infrared light. It will be directed at crystals. Yeah, you can be able to see, and it will the light produced by the crystals is around the 532 nanometer, and this is the one that we will be able to see. Well, in the case of the UV TPEF, you have this 532, the green light. It will be directed to your samples, and those which are protein will emit light at 340 or like the 400 nanometer. So this is around the UV range. So what will happen is that, let's say you have this protein, visible light, it has like this all liquid salt. When you put it under the second harmonic generations, since it's crystal, it will light. You can see that it has nice structures. And when you confirm it again with the UV-TPEF, oh, right, it's the protein that. But if you see like this, if you only have like this visible and UV-TPEF, you might say, oh, this is the protein. But actually, it does not form crystals. Because it does not form crystal, it will not appear on this second harmonic generation. Like, maybe like it will be easier like to see the real sample later. So basically, this will help you to see which one is the only the chiral crystals and the non-chiral. Okay, so once you get the protein crystals that you want, this is like the arrangement of the X-ray diffractions. So you'll get your protein like the crystal that you want. You might actually do a cryo and then you put like on loop. and you have like this is actually what we call as a goniometer. So they will like rotate, continuously rotate. So you have like a laser pointed at your rotating samples. And this is like the picture of the pattern that you will see. After you do the X-ray diffractions, and you will see this diffraction pattern. And then the crystallographer will try to see and map the electron density. So basically they start to use different methods to confirm. Like say you have like a different softwares, and then you'll see the distributions of the electrons, and then they try to visualize it with different another software again. And then after this, they usually check it again with the diffraction pattern, reconfirm, validate the result, then it will be able to be uploaded to the PDF. So that's how this actually works. And I guess, okay, so this is actually some few things that publications that uses our technology that we want to share. So this is like they try to see the microcrystallization. So they use our second harmonic generations and UPTPEL. And when they try to also see the structural basis of antibody for Zika, they also try to visualize their crystals with our technology and able to find this structure. Okay, I guess that's all from us. So thank you so much for the time.
Yes. Thank you very much, Ms. Inge. That was a really interesting topic about protein crystallography. So please, everyone, if you have like, evaluation later on, please take a look on the QR code, which has already been shared on your screen as well. So take a look on that one, use your phone, and then give some input about the topic that we have already discussed today. So I am just giving you like, from my previous experience in the past working with protein crystals. If you have already seen the concepts, or when the process begins with protein purification, crystallization, X-ray diffraction, all the way into data analysis, you notice that Ms. Inge only spent like one slide on data analysis, several slides on X-ray diffraction, but plenty of slides on number 1 and number 2. Why? Because that's where all the work, all your hard work are going to be like, be done on number 1 and number 2 when you're trying to, number 1, purify your sample. And then you have to remember when we're talking about protein crystallography, as she has already mentioned before, 95%. So 95%, it means that on your SDS page later on, when you confirm, the band should not be more than, it should not be more than 1. Okay, it should only be 1 band that appears on your SDS page, and that will be giving us the indication that, okay, that's the protein, that's the protein which is pure enough to be brought into the next one. So like if it's not 95%, what will happen? What will happen with if it's now not 95%? Then if it's not 95%, and then you have several bands on your SDS page, then the possibility of your protein crystals that you have already spent time to work on X-ray diffraction data analysis is not the protein that you're looking for. Okay, so if you have several bands on your SDS page, that means that you have protein A, right? You have protein A on this size, and then you have protein B and protein C on that appears as the 3 different bands on your SDS page. So you have already spent hours working on number 3, number 4, all the way into data analysis. And then at the end of the day, once you solve the crystal structure, you know that, oh, shoot, that's not protein that I'm looking for. Okay, so you spent a lot of time, and that's what happened to me. That's what happened to me. Just to be able to do on number 1, number 2, I spent like almost a year to make sure that my protein sample is pure enough. And then from there, then I can get it sent, doing the crystallization itself, and then try to improvise with the crystallization. So do you guys have any questions for Ms. Inge here, by the way? Or any questions for Pak Taufan as well? Or anybody from the audience on Zoom? Is there any questions over there? Okay. If there's no question, I have a question for you, Ms. Inge. Probably because I got really attracted to this machine here, and then this one is the TFF, correct?
Right. Yeah. Tangential flow filtration.
Tangential flow filtration. Okay, and then this one is just like one method of protein purification.
Yeah.
Is that correct? Yes. Okay. So everyone who's attending it today, you guys have been introduced. Probably for semester 1, you have not been introduced at all with the protein purification method. Okay, you all probably have already been shown in this presentation before that we have column chromatography. Column chromatography depends much on the gravity to pull, like, to pull the drips by drips by drips by drips that falls down. And then that drips, you need to start like, collecting them and check them on your SDS page. Is this the protein molecule that I'm looking for or not? Okay, so that's the process. However, this one is pretty new for me as someone. I've never done this like work before. Can you like, explain a little bit on this one, Miss Inge? When you're talking about tangential flow,
Okay.
I think that was a little bit on your, there was another slide over there before.
Wait, let me get back to that slide first. Okay.
Yeah.
Okay, so this tangential flow filtrations is basically like your regular filters, but it has like different size. It is in KDA or kilodalton. So it's more like for the molecule size of for you to actually filters all the molecules. So let's say here I have like a 10 KDA.
Can you put it closer to the camera or no?
All right.
Oh no. Can you point it out a little bit to over here, Pa? Sorry. So that's the filter.
Yeah.
Okay.
Okay.
Later on, for you guys who are here, you can just come over
Yeah.
here and then take a look. Yeah.
So this is actually the filters. So because let's say our protein, so this is the milk. So milk are mostly like 15 kDa above. So it's basically those kind of range. So I'm choosing 10 kDa. So hopefully that those protein will be retan. So those like all molecules like the waters and then all the fault colorings will go to the retinate site. To the permeate site, sorry. So this is like the terms maybe new for you as well. So we have like the feed. So this is like the feed that I have right now.
Which means that the mixture between
Yes.
protein, water molecules, food coloring, everything
Yes.
was before there.
Yes.
Okay.
So this is the feed, or this is like the proteins or your samples that you are working with. Okay, so it will go through, it will be like aspirated to the filters.
Okay.
Once it goes to the filters, it will like paste those membranes. So those molecules that is bigger than 10 kDa will go to the retentate side, while those with lower molecule size will go to the permeate side. So all the waters, because it is all like below 10 kDa, it will go straight to the permeate side, while those that is bigger than it will go to the retentate side, and then it will go back to this tubes. So it will, if I keep this machine going, it will like the volume will continue to like going lower, because I'm doing like this is actually a concentrating. So let's say if you want to also change the buffers, then we can change with like the DL filtrations. So we can change the buffers. So let's say when you have like a purification method, especially let's say we have like, let's say we eluded with like a phosphate buffer, which is obviously it's not favorable for crystallizations, because it's like the salt is easily precipitated, right? So you might also actually want to change those buffers with like a more suitable buffers for your protein to crystallize.
Okay. Okay. Cool. So that's just basically another method of like protein purification.
Yes.
And then in this process here, before you can like go and put your sample over here, do you need to do number one with what you call it? You don't need to do gel filtration beforehand. You don't need to do nickel NTA.
Okay. So the question is, when we do a purification, it's not a single step, of course. It is a multiple steps. So let's say, I would say this is actually, this type of filtrations is usually done, say if we have like a heterologous expressions. So if we have like a recombinant, like those expressed let's say we have an E. coli, or we have like expressed proteins in like the cells like the CHO like that, because those have like the medium. Let's say like we have like an extracellular protein. It is mixed with the medium. Then we will need to like concentrate it first, change the buffers. We can use this as the first step before we actually go straight to the purification.
Okay, so before you go to nickel NTA, for example, okay, because nickel NTA, basically the concept of nickel NTA, you guys are still like in some in semester 1, and you, Peter, are you, which semester are you? 1 as well. Okay, so in semester 1, you guys probably are not aware, but nickel NTA, it has like beads. Beads, like, like imagine a ball, like, but really tiny balls, and then it will then interact because it contains the nickel arms. The nickel arms, it's positively charged. It's positively charged, if I'm not mistaken. Anyway, I forgot that one, but it has an interaction with histidine. Okay, histidine is an amino acid which is a positive charged amino acids. Because of the interaction, that binding, it will bind to histidine, and then it will retain the protein molecule in the bits, and then later on, every single thing that is not attached to the bits, they're going to be washed away, and then things which stays in the bits will then be eluted. The process is called elution. Okay, so that's something that later, from semester 1, you guys will learn about that later on in the future. So this is a really like good thing, a fancy thing, right? Do you guys have any questions at all? Not for now. Anything from Zoom, Miss Anissa? No, at all. Okay. Next question for you, Miss. Miss Inga. What about this machine here? The basic concept of this machine here is just like depending on the UV light as well as the visible light. Is that all, or because you mentioned something on your presentation before that, for example, if there is a case that I've already found a case in the past where the protein sequence does not contain those 3 aromatic sequences. Okay, there is no tyrosine, there is no phenylalanine, there is no tryptophan. UV280, it really depends on those 3 amino acids. If there are no amino acids, no tryptophan, no phenylalanine, and no tyrosine, then UV will be useless. Okay, so can this machine do anything for protein molecules that do not contain those 3 aromatic amino acids?
So actually, the one that I'm mentioning here is like the different light configurations that we have for rock imager. So it just happened that the one that we brought is actually the UV and the visible, but you can actually have different light configurations like this MFI, or you can also have like this tonic. Yeah, it really depends on the choice of like the customer.
Okay, so the options are going to be like available over there.
Yeah.
Is it like being attached to this machine as well, or it's a completely different machine?
Different lights will need different kind of like arrangement, right?
For this for this type, so it can be applicable into this type. So for the multifluorescence of the imagers, but for the sonic, because it needs to be what's that? It's more because it has less laser technology, so it must be inside of the some storage of there. So there will be a totally different with this instrument.
Okay, cool. Thank you very much. Thank you very much, Taufan. Right. Would you like to show to everyone who is attending here about the visualization of which one is the protein molecule and which one is not? Probably if those who are attending online, if you want to see, we can just point it a little bit into the laptop.
Okay, so I'm getting help from partner here. He is our support. So and he's the one working with this RI. So basically, wait, let's start with. So what we have here is MRC plate. So this is a protein crystallized with the sitting method. And we are going to visualize it first, I think, with the visible first. Yeah. And later we will see the difference between the visible and the UV one.
If anyone wants to see what kind of like plate which is being used, I think Miss Inge has already shown. Yeah, this one here, the one to the left, the sitting drop plate. That is the exact same plate which is now being used and being put under this microscope here.
Okay, wait, let us start with the live imaging first. So this is the one that we are doing with the visible light.
Visible for those who are attending from Zoom. Miss Anissa, can you see that on Zoom? Is it quite visible? Not really. Fadil, can we zoom in a little bit closer to the camera, to the laptop over there, the screen? Right? Okay, so is that quite visible? Yes, for people who are attending in Zoom. Okay, so this one is with visible light.
Yes.
And what appears now is what, missing it?
So, okay, so what appears now is mostly like a precipitate. So this is like more like, this is like from the first, okay, the visuals that we got. This is more like a precipitance of like the buffers and all the salts here. And this is, we're still not sure. It might be a crystal, but from the ships, for me, it's not a protein crystals, but let's see under the UV light.
Now we're going to switch, click the button to UV.
So the plates over there will move under the UV light. Okay. So under the UV light, it appears quite different. You only see like some whitish spot right here. So this is actually a protein. But to further confirm if this is really a crystal or not, then we will need to confirm with other method as well.
Yes, so can you go back again, sorry, to you, visible. Take a look, everyone, to the visible one. And then you will notice, like when you see under the visible light, the structure is there. Okay, so you can really see that. Okay, there's a shape that, like, like a balls kind of shape, like rocks, right? That's the visible one, right? If you only rely on the visible one, you can actually think that, oh, that looks like crystal, that looks like rocks or something like that, right? And then however, to know whether this is protein crystals or not, you have to switch into the UV light. Sorry, Pak, can you switch again to UV light? So take a look on the difference, the main difference, if you notice it from your Zoom. Those structure is not there anymore. Okay, you cannot really see any structure that looks like rocks. And then all you can see is just like white dots and then specks on that black screen there. Those white ones is actually the absorbance. Okay, absorbance that is being result or that's being produced by those 3 aromatic amino acids. Okay, so it means that around that area there, the one which is very, very white, the protein or the amino acids, they have already formed a structure where it's mostly consisting of the aromatic amino acids. The aromatic amino acids, they are phenylalanine, tyrosine, as well as the tryptophan. Okay, so that's the main thing. When you're trying to visualize protein crystals, you have to kind of like know which one is protein crystals and which one is not protein crystal. Right. Questions from you guys? There's a question. Okay.
Okay, so there's a question from the Zoom from Nicholas. So what are the parameters that we look for in our final protein structure to decide whether it's publication worthy?
So I think Mr. Mario will be able to help me a little bit on this. But all I know about the crystallography for the publications, it also depends on the resolutions, the image. That's one of the biggest factors that we are aiming for. Because if you have like a low resolutions image, then maybe that your image or the visuals that you are getting is not reliable enough. So maybe you are getting not the real structures, maybe like this, maybe like some carbon is not like really in the place. Yeah, that might be like one of the biggest factors. Consider whether it will be like published.
Okay, thank you, Ms. Inge. And then to add on that one, guys, if protein crystals, a good protein crystals usually range from anything around 1 up to 2 point or roughly 2.8 angstrom. Okay, that's the the best range of angstrom. Angstrom is the unit size. Okay, 1 angstrom is 10 to the power of minus 10 meter, if I'm not mistaken. It's so tiny. But then the smaller the angstrom unit, then the better the mapping of the electron density, okay, of the electron that you can visualize later on, that will then appear as the structure. And it will then tell us that, okay, the structure is reliable enough. After the structure has been confirmed and verified, later on, so that's on the electron density map, the smaller the angstrom, then the denser the electrons are going to be located on the electron density map over there, right? Once you have already got a dense electron density, okay, a dense map means that electrons are sitting over here, another electrons over there, another electrons over there. Electrons, it means that there will be atoms there. Atoms means that they're going to be the amino acids that form that protein molecule, okay? So by knowing the electron density, then you will be able to tell the 3D structure, how the protein molecules have already folded, okay? And then how it has already folded, it will then tell us which one is the active site, which one is the allosteric site. If that is the active site, can you then bond the active site with any drug molecules that is compatible, that can sit on the active site, okay? That's why this protein crystallography is kind of like, important for almost everybody. The first thing, if you guys remember from COVID-19 pandemic, like everyone tried race to solve the structure of the SARS-CoV-2 spike protein. Almost all scientists all around the world. Why? Because by knowing the structure, you will be able to determine how this infection can actually occur, how the spike protein can bind to the receptor molecule, and then how the treatment, the drug treatment that will be attacked and then bind to the spike protein or any other protein in the SARS-CoV-2 virus. Okay, and then whether it's going to be publication worthy or not, it really depends on after this. After you have already generated the map using software, by the way, you don't have to do it anymore. We are in 2023, so there's a software called CCP4, and then there is online forum as well for crystallographers. If you have like difficulties, it's going to be submitted to Protein Data Bank (PDB), and then once it's being submitted into the PDB, people all around the world will be having access to that crystal that you have already generated, the crystal data. And then from there will be a verification process. And then the verification method will then determine whether your protein is going to stay in the protein database or not. Okay? Publication worthy or not, it really depends on as well the data you generate and then the way that you're presenting the data itself. Questions?
Okay, so this is like a follow-up question from Nicholas. So in part-time repositories such as PDB, they have these parameters such as Ramachandran, plot, clash score, etc. Which of these parameters should we put extra attention for publication?
Wow, that's a hard question actually. I mean, I think it really depends though on a case by case, actually, because like the Ramachandran, I think it's a bit different.
Yes, Ramachandran is one of the data that you have to make sure that you have it there. Okay, so when you before you are able to get and then submit it into the PDB, Ramachandran plot is going to be one of the data that you have to generate. That's number one. Other than that, the Fourier transform data that you generate from the protein crystals are also an important one. But then again, everything, when you reach the moment of saying, when the crystallographer says that, okay, the protein structure is solved. Okay, they call it solved. So it means that the protein data generated by the X-ray density map here has already been able to be solved by the software. Okay, the software have already positioned all the electron density according to its respective location, and then according to the mathematical equation done by the software, it's kind of like solved already. Okay, and then usually it's going to take, it can take up to as quickly as a week to solve a protein molecule, or in my case, even a year it's not solved. Okay, all right. So if you are going to talk about bad experience in protein crystallography, come talk to me. Come talk to Sir Mario. I've already got all the bad experiences from the protein crystallography, starting from number 1, protein purification, all the way until number 4, data analysis. So I know how to deal with that bad problems. Okay. Any more questions from Zoom? No? Anyone else over here? Yes, Peter? Okay, so that's a question here from Peter over here. So Ramachandran plot, if it's going to be one of the data needed, it's Ramachandran plot is basically the basis of Ramachandran plot is more about the mathematical modeling about how alpha helix and then beta sheets, as well as the 3D structure, general 3D structure of the protein molecules to be able to sit properly according to its respective location, right? Now, question from Peter here. If we just simply we have AI at the moment, artificial intelligence, which can actually do the mathematical calculation better than us, can we just simply use that one? Is that the question? Or like that, Peter? Can we just rely on that model or something like that? Yes? The question, the answer is no. All right? AI, artificial intelligence, is one thing. If you are more into bioinformatics, you're going to do online simulation. And then we have already at the moment, there is a tool called AlphaFold. So you guys who are attending it on Zoom, go check AlphaFold, ALPHA Fold. That AlphaFold is artificial intelligence from DeepMind working together with Google. They're going to just generate a model of the 3D structure of a protein molecule based on the sequence only. However, remember, it's only a model. Okay, you are only doing a calculation based on the model. The real data always comes from the wet lab activity. Right? Verification needs wet lab activity, wet lab experiments. You cannot just simply rely on modeling, because say, for example, according to modeling, it's almost 99% that this drug molecule is going to sit properly and bind properly to the protein molecules. But when you're trying to generate the data in the lab, it's not the case. It can actually be reduced up to, 95%. And will 95% be good enough for pharmaceutical company to release their drugs, okay, to cure the disease, or it's not good enough, right? So that's the answer for you, Peter. Anything else? Kenneth and Frankie and what's there? Excel. Excel is just okay, what's that? I'm just semester 1 here, and then okay, what's this? Right. But anyway, I can see that the names on Zooms, they are from more advanced level, from biotechnology, biomedicine students. So I hope that what we have already presented today will be able to kind of enrich your knowledge about protein crystallography. Right. at the end of this talk today, I would like to invite Pak Taufan as well as Ms. Inge here as representative from Formulatrix. We have a token of appreciation from our side. It's just like a certificate of appreciation. So this one is for Pak Taufan. Okay. Thank you very much, Pak Taufan. Thank you very much. And then for Miss Inge, our one and only BT graduate. Thank you, Miss Inge. All right. So, oh, okay. Okay, cool. Thank you, Pak.
Yeah, we have some small gift from Salatiga to Mr. Mario and I3L, and also we also give some gift too for Peter, giving some question to us, and then we have a 2 question also from Zoom, right, Miss? So maybe we will what is that, giving to you, and please giving to student
That's
who asked
Nicholas.
Nicholas and another one. Okay. Thank you very much, Peter. Appreciate it.
So Kenneth, do you want to ask question now to get the token of appreciation? Frankie and Excel? No?
Okay,
Okay, Pak. Thank you very much for having
Mr. Mario, thank you very
giving
much.
the talk to us.
Yeah.
And Ms. Inge, thank you very much as well. Thank you for coming back to I3L. So that's pretty much from. Okay, that's pretty much from us, everyone. Those who are attending from Zoom, thank you very much for attending it, and see you all in our next session of our talk. And for those who are attending it here, can you come over, and then we have our photo session together with the team from Formulatics. I just want to see and then take a look on the machine probably a little bit later after we take a picture together first.